Dr. Rajendar Moliani, who will speak on calcium signaling, a key orchestrator of PDAC progression, metastasis and chemoresistance. So he is an associate professor from the Regional Center for Biotechnology, Faridabad, Delhi. Over to you.
Thank you. A very good evening to everyone, and I would like to thank the organizers, Dr. Pramod Garg and Dr. Sudipta Dhar Chaudhary for this invitation, and also giving me the job to be the last presenter of the day. Our lab works on pancreatic cancer, and obviously one question that comes up when I'm presenting, not to this audience but otherwise, that why do we care about pancreatic cancer? And the data lies here, and actually this is 5-year survival data, and we are looking at the data that came from US. So for last 5 decades, what has happened that in 1970s, if anybody was diagnosed with cancer, the chances of survival for 5 years were only 50%. Now, as of today, 2/3 of patients are able to survive beyond 5 years. For some of the cancers, we have done significant progress. Almost all the patients survive 5 years and beyond. But if we look at this graph, the pancreatic cancer is the least progress has been made. 5 decades ago, only 2.5% patients were surviving, and today also it is below 10%. So certainly not much progress has been made in this area. And if we look at India specific data, and this is the recent data that came out couple of years ago, and looking at mortality trends of 23 major cancers, an important highlight of this study was that the highest annual increase in mortality is observed in the case of pancreatic cancer across both the genders. Certainly this is an area that requires attention for Indian population as well. Now our lab focuses on calcium signaling. And why do we care about calcium signaling or calciomics in cancer? Because all the key hallmarks of cancers are associated with dysregulated calcium signaling, be it proliferation, compromised or dysregulated tumor cell death, changes in the tumor microenvironment that leads to increase in angiogenesis, nutrition supply, immunodeficiency. They all in combined lead to decrease in the cancer cell death and metastasis. So a hypothesis with which we and others have been talking about for last 10 years is that when normal cells, they transform to cancerous cells, there is actually a change in the expression of calcium pumps, channels and transporters present on either plasma membrane or intercellular organelles. And this leads to either transient rise in calcium, a sustained increase in the calcium, or calcium oscillations. And these different sort of signals are sensed by different effector proteins, thereby leading to a different outcome. Interestingly, there are drugs out there that can act on these receptors, whereby we can regulate these signals. Now the key point is to identify these molecules or these transporters or pumps that can be uniquely targeted for managing pancreatic cancer. So we started looking at the expression of these calcium channels and transporters in publicly available data sets like TCGA and GPA. And we got several hits. Today I will be focusing on only one channel, that is ORI3. And what we found that in comparison to normal pancreas, in pancreatic tumor samples, ORI3 is overexpressed, and this higher expression of ORI3 leads to poor survival. All the patients that have high levels of ORI3 die within 6 years of diagnosis, whereas a proportion of around 30% of patients which have low ORI3 levels are able to survive beyond 8 years, suggesting that higher ORI3 levels are associated with poor prognosis. So we started studying this. We have done lot of work on ORI3, but to introduce to the audience what is ORI3, Joydeep just talked about ORI1. So essentially these are store operated calcium channels which are activated when growth factors binds to the GPCR receptors, thereby leading to generation of IP3. IP3 goes and binds to IP3 receptor which is present on the ER. Calcium from the ER comes out to the cytosol, and then this leads to decrease in calcium levels within the ER. Over here I would like to highlight this sensor protein STIM1. Calcium is typically bound to it, and it is like a compressed spring. When calcium levels are decreased, this compressed spring opens up, and now this opened spring interacts with the ORI channels present on the plasma membrane, thereby calcium coming into the cells. This calcium then drives physiological functions and contributes to cancer progression. We have done lot of in vitro experiments, but in the interest of time, I'm just showing the in vivo data over here. So what we did, we injected cells, pancreatic cancer cells, expressing GFP, and these are control cells and either cells which are having shRNAs targeting ORI3, thereby decreasing the ORI3 levels, and we looked at tumor growth. And as you will appreciate from these pictures, when we decrease the ORI3 expression, the tumor weight, the size, weight and the tumor volume, the growth overall decreases substantially. Since we had injected cells which were carrying GFP, the protein which is typically not expressed by mice, we were able to perform bioluminescence studies to look at GFP expression within mice, and that gave us a surrogate for studying metastasis. So if you look at, this is the control condition and this is the ORI3 silence condition. In the control mice, there were lot of metastases that could be seen here, and the tumor size were also higher. But in the knockdown condition, you will appreciate that the tumor metastasis was either absent or very less. So collectively this work is published a few years back. I won't spend much time on it. Essentially the summary is that in pancreatic cancer cells, ORI3 is hyperexpressed, overexpressed, and that in turn contributes to tumor formation and metastasis, and this overexpression is associated with poor prognosis in patients. Now I will switch gear and little bit talk about gemcitabine resistance. Gemcitabine is the first line of therapy for pancreatic cancer, and if we look at if there is any connection between gemcitabine resistance, chemoresistance and calcium signaling. So there are a couple of studies that have looked into what sort of proteins are getting enhanced when there is gemcitabine resistance in pancreatic cancer cells. Interestingly, a protein that comes up in this is STIM1. And if you remember, I talked about STIM1, it is the sensor protein, the compressed spring, which opens up and activates ORI channel. So what they showed that STIM1 is overexpressed along with RRM1. So RRM1 is the hallmark of gemcitabine resistance, and interestingly they are present at the same genetic locus on chromosome 11, and a more detailed study showed that when STIM1 and RRM1 are overexpressed in the gemcitabine resistance condition, there is more calcium in the cells, and this calcium drives the gemcitabine resistance. An interesting question is, although they said that there is more calcium which is coming from the outside, they did not identify the channel which is responsible for bringing this calcium into the pancreatic cells. With this background, we started studying gemcitabine resistance, which is a big clinical problem. We generated gemcitabine resistant pancreatic cancer cell lines, and then we looked at ORI3 levels and contribution of ORI3 in gemcitabine resistance. What we found that ORI3 is overexpressed in these gemcitabine resistant cells, and if we knock down ORI3 in this gemcitabine resistance background, we see that affects the chemosensitivity. And what do I mean with this? If you take the parental cell line, the IC50 of gemcitabine is around 2 micromolar. If you have the gemcitabine resistance, this IC50 goes up to 35 micromolar. You have a control which is also around 36, 37, and if you knock down ORI3 in this gemcitabine resistance background, this IC50 again comes back to 1.5 to 2. That means ORI3 is in the gemcitabine resistance case providing this resistance. We also did more detailed apoptosis studies using flow cytometry, and essentially we got the same data. The apoptosis is higher in the parental cells. In gemcitabine resistance it is decreased, but if you knock down ORI3 in this resistant background, the sensitivity or the apoptosis again increases, suggesting that ORI3 contributes to chemosensitivity in gemcitabine resistant cells. We did variety of assays, like this is migration, and we also did invasion. Migration increases in gemcitabine resistance background, and if in this background if you compare the control or ORI3 knockdown, this migration capabilities of the pancreatic cancer cells are substantially reduced. We looked at 3D spheroid assays because they are the real hallmark of stemness and also chemoresistance, and again essentially we saw the same outcome, that in gemcitabine resistance there is increase in stemness, and this increase in stemness is decreased when we knock down ORI3 in this background. So now this was all in vitro. Now we wanted to see if ORI3 actually plays a role in gemcitabine resistance in vivo, and a good readout is increased metastasis, because whenever there is a relapse of pancreatic cancer, it is also associated with metastasis. So we use zebrafish as a model system, and when we inject the cells in zebrafish, we can study them. If they are tagged with a fluorophore, we can study their migration and metastasis. So using this, we carried out injections with the parental cells, gemcitabine resistance and ORI3 knockdown. And as you will appreciate, and the data is quantitated here, gemcitabine resistant cells, they metastasize more, but if you knock down ORI3, the metastasis decreases. To understand the molecular mechanisms, we carried out transcriptomics studies, RNA-seq on all 4 conditions, parental cells, gemcitabine resistance, control and ORI3 knockdown. And essentially what we were looking for, the signatures that are going up when cells are acquiring gemcitabine resistance, and at the same time they should go down if they are working downstream of ORI3. So essentially things that are going up in gemcitabine resistance versus parental, and going down when we knock down ORI3. And when we did this, we found 12 genes. But the student was, bit hesitant to work on 12 genes. So she was smart and she overlaid this data with what is database that is available, calcium gene database. And when the overlay was done, there was only one single hit that came out, which was SLIT3. So what is SLIT3? SLIT3 actually works with robo, and they are known to be functioning in exosome signaling. And when we looked at literature, interestingly there was some data from Australian Pancreatic Cancer Genome Initiative under the leadership of Dr. Seen, and what they had reported in 2 independent nature studies, that SLIT and robo mutations were reported, and these mutations were associated with pancreatic cancer, higher pancreatic cancer risk. And interestingly, SLIT3 was also shown in case of hepatocellular carcinoma to be involved in chemoresistance. So that means SLIT3 could be an interesting target working downstream of ORI. We validated that, we knocked down SLIT3. Knockdown of SLIT3 decreased the resistance, as you can also appreciate from the 3D spheroid assays, migration assays. So essentially downstream of ORI3, SLIT3 functions, and thereby it regulates gemcitabine resistance. More details of these studies can be found out on a paper which is now available on bioRxiv and under revision for a reputed journal. So to summarize, in pancreatic cancer cells, ORI3 is overexpressed. That leads to pancreatic cancer progression and metastasis. It contributes to pancreatic cancer chemoresistance. We also have some data on partial EMT, role of ORI3 in that, but in the interest of time I am not discussing it, and this partial EMT actually contributes to both higher metastasis and chemoresistance. Finally, the future directions, what we are doing right now, since in patients the ORI3 is overexpressed at mRNA level, we are trying to understand transcription regulation of ORI3. We are getting into more detailed molecular mechanism, and with the help of molecular docking and in silico screening, we have identified some therapeutic drugs that can act on ORI3 to control the disease progression. And some of the transcription data, I did not got time to discuss this, has been just accepted in EMBO journal. It should come out in couple of weeks, but the preprint is available again if someone is interested in studying this. With this, I would like to acknowledge the team behind the work, collaborators. The drug screening work we are doing with Shailendra Asthana and Dr. Pramod Garg at AIIMS, and lab is currently funded by almost all leading agencies of India, as well as international funding from EMBO and India Alliance DBT Welcome. With this, I would like to thank you all for your attention, and will be happy to take any questions.